Okay. So hello everyone. So it's my great pleasure to share the research work done by my team in the past 10 years. Actually started from my PhD work. So the title for my today's presentation is the Microbial-mineralization in soil, applications in geotechnical and geoenvironmental engineering. So firstly, I would like to thank all my current and previous team members. So without their hard work, it's impossible to get all this hard work done. And I also like to thank these funding agencies. So only with their money, then it's possible for me to do the work. So for today's presentation, so firstly, I would like to briefly, go through the background. As you can see, more than 1/3 of audience today are quite familiar with MICP, so that's very good. So I will just spend a little bit of time here. Then after that, I will introduce my work in 3 different periods. The first one, MICP for soil hydraulic erosion control. For that work, I did it during my PhD study. And for second one, MICP for calcareous sand stabilization. That's my work when I was still in US, in Hawaii. And for third one, the MICP for heavy metal immobilization in sandy soil. That work was, my current work. Then finally, I would like to spend a little bit of time to talk about the future of MICP, my own thoughts. Okay, so firstly, as we can see in the classic soil mechanics, so there's only 3 phases for soil: the solid, water, and air. It means that we regard the soil as an inorganic material, right? And all current, theories are based on the inorganic material. But actually, as we all know, right, soil contains a lot of things, other than the solid, water, and air. The most important thing actually is different kind of, biological things. The larger one like the plants, the animals, the small one like the bacteria, the fungi, even the virus, right? So we have, a variety of, biological things within the soil. But unfortunately, in the traditional soil mechanics framework, we don't take them into consideration. And from other fields like the environmental engineering or the agriculture, then we know that actually the biological activity could, affect the soil properties a lot. Not only, in their aspect, but actually also in the mechanical, hydraulic kind of things. And that's the things that we care about as a geotechnical engineers. So based on this background, so especially in the past, I think in the past almost 20 years, geotechnical engineers are looking at, different biological activities in soil, in particular, the microbial mineralization, or sometimes also called biomineralization. So biomineralization can be triggered by many different, processes. For example, the oxidation reduction, or the photosynthesis, or the MICP, as we talk about it. So for MICP, I think the process, I guess most of you are familiar with it. So for MICP, when we talk about MICP, we normally refer to the ureatically based MICP. So in that process, the urease producing bacteria could produce the urease enzyme. This special enzyme hydrolyzes urea to produce ammonia and carbonate. And during this process, the system pH also increases. So then if we also have the calcium ions present in the system, then the calcium can react with the carbonate ions to form calcium carbonate precipitation. So when in soil, then the calcium carbonate precipitation can serve as a cementation or cementing agent, so they can cement particles, soil particles together. So this is something we call the binding strength formed by the calcium carbonate precipitation. And actually, this is the primary mechanism of my MICP for granular soil stabilization. Then in addition to that, so the formation of calcium carbonate crystals within the soil can also improve the overall density of the soil. So this can also contribute to soil strength. And finally, because some calcium carbonate crystals also serve as a coating, on the surface of soil particles. So this can change the particle roughness. So all the three, mechanisms, overall contribute to the strength enhancement in granular soil by MICP. Okay, so next I would like to briefly talk about the strategies to implement MICP. So here, as you can see, there are three, primary methods. The first one, bioaugmentation. So in this method, the bacteria come from the lab, right? So usually we purchase some kind of standard uritic bacteria. Then we cultivate it in the laboratory. When it reaches a certain concentration, we can inject the cultivated bacteria into the ground. So for this method, obviously the disadvantage is that the compatibility between the injected bacteria and the native condition may be quite bad. So actually, if I remember correctly, there's one previous research, looking at it, only after like 1 or 2 days, all injected bacteria are replaced by native bacteria. So I think that's the limitation for this technique. But the advantage is that it's easy to control if we use standard bacteria. I think that's the advantage. So the second one is something I call the ex situ biostimulation method. So in this strategy, so firstly, the soil samples are taken from the field. Then in the laboratory, the researchers can do the purification, isolation, and extraction of the indigenous bacteria. Then the enrichment is performed so that we can get high concentration indigenous bacteria solution. Then the solution is re-injected into the ground to trigger the MICP reaction. Compared with the bioaugmentation process, obviously for this one, the compatibility with the native condition is much better. But we can also say that if this strategy is used, then the whole process is quite lengthy and it's quite complicated. And we need a mycologist to involve in this process in order to get indigenous bacteria selected and enriched. For the third one, the in situ biostimulation. For this one, instead of taking soil back to the lab and do the extraction, isolation, and purification, then the enrichment medium is directly injected into the ground to stimulate growth of indigenous bacteria. For this strategy, although we don't know what exact species has been enriched, we don't know that unless we do some testing, right? But that's fine. What we want to achieve is the uritic activity, right? So with this method, we can easily make the suitable bacteria to grow and achieve, satisfactory uritic level so that we can do MICP. So in my research, when I did my PhD at Cambridge, I focused primarily on the bioaugmentation because that's, at that time, many people simply did it in this way. So I just followed them. But then after I joined the University of Hawaii, because Hawaii strictly restricts the use of bacteria to do the test, especially in the field, I have to switch to the biostimulation strategies. That's why for my later study, I mainly focus on the in situ biostimulation strategy. Then for MICP research at different scales. So currently, there's numerous studies at all these, scales, from nano to micro to element scale to pilot scale to field scale. So for example, at nano to micro scale, people have used like SEM to see the microstructures, use the atomic force microscope to see the surface morphology at nano scale, use the microchip to see how bacterial solution and the cementation solution migrate in the porous medium. At the element scale, all the conventional soil testing methods have been used, like a traxel shell test, the one-dimensional compression test, sort of things. And at pilot and field scale, there's also many reported studies. On this slide, I just show some representative works from these famous groups. For example, Jason Ning-Jun from UC Davis, use it for the surface erosion control at a desert area in the US. Professor Jiang from Nanyang Technological University in Singapore use it for the sealing of the inside part of a pond for sealing purpose. And Professor Liu Hanlong from Chongqing University in China, he used it to enhance sandy foundation in the island. So it's just like making artificial island using this technique. Professor Van Passen, I think he returns to Netherlands now. He used this technique for liquefaction control. Professor Kawasaki from Hokkaido University in Japan, he used it for the slope stability control. Professor Kabazenjin from ASU, he used it to make the soil column, as you can see here, it's quite strong, like a stone. And Professor Miao, also from my university, used this technique for the wind erosion control in desert area in northern part of China. Professor Montoya from North Carolina State University, she used it for the coastal erosion control. And Dr. Sania used it for the control of contaminants. So these are some representative work done by the famous groups worldwide. So for my group, so we published 3 papers from 2019 to 2022, talking about the future, the state of art, and in particular, the application of biomineralization in environmental geotechnics. So if you are interested in this content, please feel free to check these papers. Okay, so next I will move to my work as a PhD student at Cambridge. So at that time, if I remember correctly, in the UK was suffering from quite large flooding at that time. So my supervisor, Professor Soka, just mentioned, okay, do you want to combine your bacterial technique with the flooding control demand in the UK? Yeah, I think that's a good idea. So that's why I go here. So for the hydraulic erosions, as we all know, there's various kind of forms, like the dam internal erosion, like the seepage piping in earthfield dams, coastal erosion, or rainfall-induced erosion, many different forms. So in order to understand the fundamentals of hydraulic erosion, as well as find a suitable control strategy, it's important to understand that, firstly, soil properties like the particle size distribution or fine content could affect a lot of the erosion potential of the soil. Secondly, the hydraulic induced soil erosion is quite a complicated phenomenon. It's a mechanical hydraulic chemical biocoupling process. So you have to understand all these aspects. And finally, for different mitigation methods, we need to find a suitable way to assess the erosion control effectiveness. So bear these questions in mind, then I come to my research. The first one, I looked at the internal erosion control of grass sand mixtures. So they are both gravel and sand are granular soil. So it's granular soil-based problem. So in this research, so I developed a large one-dimensional column internal erosion testing apparatus, and this one was combined with an MICP implementation unit. So I can prepare the sample within it and then implement the MICP to treat it. Then from the figure on the right-hand side, as you can see, the first line shows the column without MICP treatment, and the second line shows the soil with MICP treatment. So obviously, when the hydraulic pressure is like 30 kPa, then the non-treated samples, in the non-treated samples, we can see a lot of erosion on one side. But for the MICP treated samples, no erosion at all. So that's quite evident from the visual observation. Then I did a further investigation. Firstly, I looked at how MICP cementation solution concentration could affect the efficiency. So the left-hand side figure shows the erosion rate versus time, and each color shows one MICP cementation solution concentration. And the second figure shows the erosion rate versus the flow velocity. So from this figures, we can see that the treatment with only 0.2 mole cementation solution could already reduce the ultimate erosion percentage to less than 5%. With higher cementation content, we can almost say no any erosion. And also, if we talk about the critical flow velocity, which is the turning point in this graph, the MICP treatment could also increase the critical flow velocity significantly. In addition, I also looked at how original soil composition could affect the erosion. So here I look at two structures. The top one is the gravel supported structure, and the bottom one is the sand supported structure. So for the gravel supported structure, so we find that MICP treatment could very successfully mitigate the erosion. So like the dotted line showing in this graph, and it also only marginally affected the permeability, as you can see here, because here the gravel is a skeleton, so the permeability is controlled by the gravel. Therefore, the sand supported structure is quite complicated. MICP is not always effective, especially when the cementation level is not high. So it means that here we have to apply higher cementation solution concentration in order to make it more effective. So that's the first part of my PhD research. The second part, I moved to the sand clay mixtures. So sand is granular soil, clay is fine soil. So it's fine granular soil mixture. Okay, so here in order to investigate the sand clay mixture, firstly, I developed another testing apparatus which features the turbidity meter. So the turbidity meter here is used for the clay concentration measurement, and the erosion is obtained. Erosion rate is obtained based on the data from the turbidity meter. Then the right-hand figure shows the peak erosion rate versus shear stress. So here we can see after MICP treatment, firstly, the turning point or the critical shear stress increases. Secondly, for the post-erosion part, the slope for the post-erosion part, which we call it erosion coefficient. The erosion coefficient also drops after MICP treatment. So this can demonstrate that MICP treatment can also mitigate internal erosion in sand clay mixtures. So then we also look at other hydraulic erosion forms, for example, the sand dune erosion. So in this model test, we consider several conditions like, for example, the slope degree, the 22, 26, 30 degree. We consider different magnitude of wave, small wave or strong wave. So from this model test, we can see that under all these conditions, either small or strong wave, either 22 degree or 30 degree slope, then MICP treated case performs the best. It's even better than the EICP case and much better than the control case, which is without MICP treatment. Using the same model test apparatus, we also perform the rainfall erosion test. In this case, we use the new method, a photogrammetric approach based on the structural motion or SFM to get the erosion on the slope surface. So as you can see on the right-hand side, so the blue indicates erosion and the red indicates accumulation. So for the top one, it's untreated case. So we can see, quite uniform erosion on the surface of the slope. Then for the MICP treated case, so only a little bit erosion in the middle and some accumulation at the toe part of the slope. So this result could also demonstrate the effectiveness of MICP for the slope surface protection. Okay, so the next part, I would like to talk about my work, in US, in Hawaii. So because in Hawaii, we have calcareous sand surrounding all islands, not only Oahu, but Big Island, all other islands. And unfortunately, as I mentioned, we were not allowed to use cultivated bacteria from the laboratory. So we had to switch to the biostimulation strategy. So here we prepared enrichment medium consisting of either ether extract or nutrition brass plus urease. The cementitious solution consists of calcium, urea, and some trace amount of other nutrition additives. So firstly, the enrichment period is 48 hours by surface percolation. After that, then the cementation solution percolation once a day for 5 to 10 days. Then we can make the cemented soil sample. Then after the first stage, we took some samples and measured the radiative activity, pH value, urate concentration, and also some genetic analysis. For the biocemented sample, we measured the cementation content, its microstructure, and we also performed the conventional soil testing like the direct shell test, traction shell test, and odometer test. So firstly, the genetic sequencing results. The first column on the left side shows the original soil. As we can see, the distribution of different species are quite uniform. We can see a variety of species. But after stimulation, either by ether extract or by nutrient brass, we can see some dominant species, like the blue one, which is the Sporosasana, or the pink one, which is the Bacillus. Both are euleritic bacterial species. So this result can prove that after stimulation, some euleritic bacterial species become dominant in the system. So that's good for us. Then we used SEM. For this one, we used the microscope to see the microstructure of biocemented soil. The figure A shows the control sample without cementation. Figure B shows the sample with only a little bit cementation, 1.8% cementation. So in this case, we can see some cementation connecting soil particles, but not a lot. Then for C and D, they are heavily cemented case. So we can see quite a strong interparticle cementation here, and also some calcium carbonate crystals exist on the surface, serving as a coating for the sand particles. Okay, so then the direct shell test. So here we looked at the relationship between the peak shell stress and the cementation content. So obviously, with the increase in cementation content, the peak shell stress increases. And this is especially evident when the normal stress is small. But if the normal stress is quite high, like 600 kPa, then this increase becomes insignificant. It means that high normal stress suppresses. Then, based on the direct shell test, we also get the cohesion, and here it shows the cohesion versus cementation content. So again, cohesion increases with the cementation content. As you can see, this dots here, and it's even better than some previous work who used the bioaugmentation strategy. Here is the one-dimensional compressibility test. So with the increase in cementation content, firstly, we can see the compression curve moves upward. Then secondly, we computed the coefficient of compressibility at different stress levels. So for all stress levels with the increasing cementation content, the coefficient of compressibility, drops. It means that MICP treatment could improve the soil's resistance to compression. Then for the CD traxel shell test, so first the graph shows the samples on the 400 kPa and 50 kPa. For 400 kPa, we can see some shell band here. For 50 kPa, it's just like a barging failure mode. Then the second graph shows the stress path, deviator stress versus effective mean stress. Because here it's a trend test, so it's just a straight line. And but the peak point different. For the solid line here, it's biocemented case. For the dotted line here, it's uncemented case. So the biocemented case actually basically has higher peak value than the untreated case. Then based on the stress path result, then we can obtain the failure envelope, right? So here shows two cases. The black one is with moderate cementation, 4.1% cementation content. And red one shows heavy cementation case with 9% cementation. So compare these two, we can see that their cohesion is different. For 4.1, cohesion is 5.82. And for 9% cementation, the cohesion increase is 41 kPa. So cohesion increased significantly. But for friction angle, effective friction angle, it's largely the same. And for the residual shell strength, so cohesion, all comes back to zero. It means that all cementation has been broken. Then the residual friction angle also largely independent of cementation level. Next, for the CU traxel shell test, so again, on the 400 kPa confined pressure, we can see clear shell band. For 50 kPa case, it's barging failure. Then for the deviator stress, effective mean stress relationship. So compared with the untreated case, which is the dotted line, the MICP treated samples exhibits the dense sense characters. It moves to the right. And it also shows stronger dilatancy compared with uncemented case. Then if we look at the deviator stress versus axial strain relationship, so when the cementation level is high, for example, the dotted lines here, then the stress is also higher compared with the lower cementation content case. And in this case, we also observed the strain hardening phenomena. Okay, so to this point, we already demonstrate that MICP, biostimulated MICP treatment could improve the strength of calcareous sand. Then we think about more. Can we further increase the strength while also address the ammonia problem? Because as we know, ammonium is the byproduct of the MICP reaction. And unfortunately, most researchers don't focus on this aspect. So here we just think about it: improve strength, reduce ammonia. So then, actually, my PhD student, think about using biochar for this function. So the first graph shows the cementation content versus biochar content. In 4 separate cases, higher biochar content is corresponding to higher cementation content. So it means that biochar can help improve cementation. So that's good news for us. Then we did the traxel shell test again, but with the addition of biochar. Then we got the failure envelope, and based on failure envelope, we got the cohesion, peak friction angle, and the residual friction angle. So here we can see that. So for the pink one and the light blue one, they are with biochar. and the blue one is like a biosimmetric case without biochar. Then we can see both cohesion, peak friction angle, and residual friction angle are increased after the addition of biochar. Then if we compare the pink one with the light blue one, so their difference is the morphology or the shape of the biochar. The light blue one is a powder shape, and the pink one is like the larger one, like the needle one. Yeah, and obviously, if we have finer biochar particle, then its enhancement effect is better. Then we also look at how the concentration of ammonia can be reduced through biochar addition. So for the blue one here, that's with the fine particle biochar. Then we can see when we do the flushing, then the ammonia can be absorbed by the biochar, so it reduces the fastest compared with the other two cases. Okay, so then based on this research, we proposed three mechanisms of biochar enhancement for the biocemented calcareous sand. The first one is that biochar can provide more nucleation sites, so we can see more cementation. Secondly, with the addition of biochar in soil, then the overall density is improved. That's two advantages. The third mechanism, which is a disadvantage, because the strength of biochar itself is quite weak, it's quite small. So when they are in the soil system, they are kind of weak points. So when we do the sharing, do the compression, then the soil failure usually starts from these weak points. Okay, so that's three mechanisms overall contribute to the biochar function in improving the mechanical performance of MICP treated calcareous sand. Then finally, based on all this research, we proposed a stress model for the MICP treated, including the biochar enhanced MICP treated calcareous sand. So as we all know, based on the Bolton theory for sand strength, the shell strength consists of friction, dilatancy, particle breakage, and rearrangement. So that's the black lines. So then with the MICP treatment, so we have additional contribution from cohesion. So as you can see, the red one, right? So the cohesion contributes to this part, especially when the effective normal stress or confining pressure is small. So this effect is quite significant. But at higher normal stress or confining pressure condition, as we can see from the direct shell, traction shell test results, then the contribution of cohesion is diminished. So it comes back to the original failure envelope. Then with the help of biochar, firstly, when the normal stress is small, it can further increase cohesion because it can generate more cementation. Then even if when the cementation is broken, when the normal stress or confining pressure is high, it can still, above the original failure envelope. So as we can see here, right, for the residual friction angle, the red one, it can also improve it based on the original, compared with the MICP treated sample without biochar. So for this one, we guess that's mainly because of the function of biochar to modify the sand surface. So it increases the friction between the sand particles. So that's why we see the failure envelope moves upward a little bit. Okay, so that's the framework we proposed. Okay, then next one, I would like to talk about my current work here in China. So my team use MICP to treat heavy metal, in particular in sandy soil. So in order to make it possible to use MICP to immobilize heavy metal, firstly, we need to understand, heavy metal species, how heavy metal species and concentrations impact the MICP. Then secondly, we need to see how, urolitic bacteria, either from the laboratory or from the field, respond to the heavy metals. And also we need to understand the immobilization process at different scales. Then we, did different tests like stress test and toxic leaching test to see, after MICP treatment, how soil properties are changed. Then in order to understand the mechanisms, we look at the contaminant phase evolution, as well as we propose immobilization model. Okay, so this slide shows the research method. So in this particular test, we use 90% sand plus 10% kaolin to simulate a clay sand soil. Then the clay sand soil is mixed with artificially prepared lead contaminant to make the artificial contaminated soil. Then the artificial contaminated soil is mixed with enrichment and cementation solution to trigger the MICP process. And finally, we can make the kaolin sample. For one part, they are subject to the viable cell number counting, as well as the measurement of urea concentration. For the other part, we did the stress test, toxic leaching test, and permeability test. So this graph shows the viable bacterial number as well as the urolitic activity results. For graph 1, the cell number versus time, as we can see, the black one is the baseline, then 20, 30, 40, 50, 100, they are the contaminant concentration. So obviously, with higher contaminant concentration, then it becomes more difficult for bacteria to be stimulated, especially when it reaches 50 or 100 millimole. It's almost impossible for native bacteria to be stimulated. Then, if we look at the urea concentration versus time or ammonia concentration versus time, we can see without contaminating the system, it can be decomposed very quickly. Then even with like 20 millimole lead in the system, then this decomposition rate becomes much smaller, but it keeps going. That's good, it keeps going. Okay, so then because we hope that after the treatment, this site can meet the environmental regulations, so the toxical leaching has to be done. So here we did a test under 3 different conditions: the neutral condition, the weakly acidic condition, and the strongly acidic condition. For the neutral condition, so as we can see, with the increase in curing time, right? So after 14 days of treatment, so the leaching concentration mostly can reach the standard set by the China Department of Environmental Protection, which is 0.1 mg per liter. And the only exception is the 100 millimole case, because the initial concentration is simply too high. Therefore, with weakly acidic case, it's still okay, still no problem, unless for the 100 millimole case. But unfortunately, for the strongly acidic case, here we use the acid of pH 2 to do the leaching. Then unfortunately, in all cases, they cannot meet the regulatory requirement. So it means that if we want to use it in the field, we have to be very careful about the groundwater condition. If it's acid, then we have to do special protection. Then we also look at its stress property. Here we performed the drain shell test. So here I just show one example, right? The solid one shows the case, the baseline case, the MICP treated sample without contaminant. And the dotted one is with 20 millimole lead in the system. So here we can see with only a little bit contaminant in the system, the stress actually increases. I think that's to the surprise of many people, but that's the phenomenon we observed in the lab. We guess it's because with only a little bit of contaminant in the system, it can help to further, stimulate the bacteria to work, because they have to survive, in this kind of contaminated condition. So they have to work hard. So they produce more urealytic urease enzyme. So the cementation content is also higher. So actually, we also measure the cementation content. It's similar to the stress result. Okay, so next, in order to validate its suitability, in the field condition, we designed a small model test apparatus to simulate a radial injection condition, as you can see here, right? So here we have a cylindrical vessel. Then we have one layer of the paper here. Then it's the compacted clay as a sealing layer, followed by HDPE here. Then it's our 60 millimeter target soil. It's also sand clay mixture. Then finally on the top, it's another 20 millimeter compacted clay layer to prevent the preferential flow between the sand clay mixture and the top layer. Then we do the injection of both enrichment solution, as you can see here, and the cementation solution from the perforated tube in the center. So then we take, the solution samples from various locations, as you can see the small dots here. So in this graph, it shows the evolution of solution pH, urea concentration, and viable cell number, at different time. The first line shows the values after the first enrichment, and the bottom line shows the value after the final enrichment process. As you can see, with more phases of enrichment performed, then the solution pH becomes more alkaline. And for urea, initially there's still a lot remaining, but after many phases of enrichment, only a little bit left. For a viable cell number, again, initially it's only like around 10.6 CFU per millimole. Then after several rounds of enrichment treatment, so for the center part, the viable cell number concentration can reach 10.9 CFU per milliliter. So this model test can clearly show that the biostimulation works in this case. Then we also take samples after the treatment to do the leaching test, and the results are also quite good. For two cases, then the remaining the leaching concentration is only between 0.1 and 0.01 mg per liter in these two cases. It means that the immobilization efficiency is quite good. Okay, then finally, in order to understand why MICP can immobilize the heavy metals, so firstly, we did the sequential extraction test to analyze the phase distribution. So based on the sequential extraction theory, so there's 5 different phases: the exchangeable, carbonate bound, the firm manganese oxide, organic bond, and the residue. So this one is the most unstable one, and the residual one is the most stable one. So after 1 day treatment, as we can see, most are in the exchangeable state, more than 70%. But when it comes to 14 days after treatment, then the proportion of exchangeable state, the green one, is reduced to less than 1%. Then most are in the carbonate form. It means that we have carbonate, in this case, carbonate lead. So it shows the immobilization results from the phase distribution aspect. Then we also used the SEM to see the microstructures and the crystals. So for the case without the lead contaminant, we can see calcium carbonate. Then for 20 millimole case, we can see both calcium carbonate and lead carbonate. And from the EDS scanning, now we can see many bright spots showing the crystal of lead and the calcium. Then if it comes to the worst case, with 100 millimole lead, we can see only a little bit bright spots and almost no bright spots for calcium. It means that the crystals cannot form very well in this case. Okay, so here are a list of the published papers from our research group, ranging from my PhD period to the present time. So if you are interested, please feel free to read this paper, and any questions are welcome. Okay, so finally, before I conclude today's presentation, I would like to spend a few more minutes to talk about the way forward for MICP. From my aspect, from my opinion, so in order for MICP to be used in the field in large scale, we have to address these three aspects. Firstly, we need to further our understanding of fundamentals, for example, the microbial processes, the chemical processes like the hydrodynamic of the crystal formation, like the interaction between the crystals, the sand particles sort of things, and also the biochemical fluid in porous medium. Yeah, that's something we have to understand in order to better control the parameters. And secondly is the multifunctionality. So MICP not only works for soil strengthening, but also it can help to solve like the environmental ecological problems. Like in the three case I showed to you, it can be used for heavy metal immobilization. In addition, it can also used to reduce like carbon footprint, can help to preserve like the water, preserve water environment, help to protect the ecological environment. That's something we can explore so that its application in the future can be wider. Finally, we need to be careful about the sustainability of this technique. For example, if we use biostimulation in the field, where it severely affected the native, biological species, when we stop it, can it recover to its original state? We don't know yet. Secondly, for all the raw material we use for MICP, are they environmental friendly? That's something we have to check. Thirdly, how about its long-term performance? As I mentioned, right, where we have acidic, even if a little bit acidic groundwater, for a long time, it's dangerous for the bio-cemented soil. And finally, how about its embodied energy? It means that is this process itself, carbon neutral, or it still consumes a lot of carbon, right? That's something we need to think about. Then finally, I want to spend 1 more minute to make an advertisement for this journal, the Geotechnics. This is a quite new journal launched last year, and Professor Nanjing is the honorary editor-in-chief for this journal, and Professor Liu Hanlong and Professor Jiang are editor-in-chief. So this journal now has been abstracted and indexed in Scopus, GeoRef, NASA ACE, Google Scholar, etc. And probably this year, next year, it can be indexed by SAI. That's good news for us. And all different types of submissions, research article reviews, editorial technical notes, case reports are welcome. Now, even if your submission is not very closely related to biotech, it's okay, as long as it talks about the relationship between, biological activity and the Earth's material. I think that's okay. So your contribution is greatly welcome at this stage. Okay, thank you. That's my presentation today. Thank you.
Yeah, thank you so much. Yeah, a lot of excellent research, fundamental research, very detailed research. So I'm sure, more research is coming up, more papers are coming up from your group. Looks like a lot of interesting work that's going on. So I'm very happy that you are able to share your current research, or even research that you are research that is coming up in your future publications. So it's really nice to see
Thank you.
Yeah, some of the new research that's coming up.